Hi everyone, welcome to this video on transcription. And transcription is going to be the first step of protein synthesis, and we've got one major goal that we're trying to accomplish by the end of transcription. And that goal is to take our DNA and to make a copy of it in the form of a molecule called RNA. And so it's going to be a really similar kind of molecule. They're both nucleic acids, but we're just going to be making a copy that's going to be able to go out into the cytoplasm, and we're going to be able to use this RNA molecule to build our polypeptide strand. So the rest of these slides will just be a little bit more of a specific details about how this process of transcription occurs. And the main steps are on this page, and there are three sort of big steps that happen. So the first step is that the two DNA strands have to separate apart from one another. So that's shown going from here to here. Normally our DNA is in that double helix form in the nucleus, but the two strands are going to separate similarly to how they separate in DNA replication. One of the strands is going to be used as a template to build an RNA molecule. So in this picture, the bottom strand is acting as a template for this growing RNA copy. And then our last step is that RNA polymerase is going to be the enzyme that links RNA nucleotides together to build our final finished RNA transcript. So we'll go through each of these steps in a little bit more detail. But first, I want to just have us remember how DNA replication worked, because the process for transcription is going to be really, really similar. So the idea for DNA replication was that with just one piece of DNA, one strand of DNA, you could rebuild the other strand. And that's because of these complementary bases. So we knew that whenever we had a C on this strand of DNA, there had to be a G on the opposite strand. If we had a T or an A on this strand of DNA, we had to have a T on the opposite strand. And similarly, a T would mean that there's an A opposite, and a G would mean that there's a C opposite. So if you had just one strand of DNA, you could rebuild the entire other strand. And the other piece of information that I want us to remember from DNA replication is that our enzyme was called DNA polymerase. And that's because we were building a DNA strand using DNA as a template. So with those two pieces of information refreshed in our memories, let's look at this image of RNA polymerase making an RNA copy. So similarly, if we have just one strand of DNA here, we're looking at the top as the template. This piece of DNA says ATG as the first three bases, the first three nucleotides. And usually if we see an A on one strand of DNA, we would know that the other strand needs a T. However, since RNA is a little bit different than DNA, remember it doesn't use T's. Instead, it has a U. So instead of adding a T to go with this A, we're going to add a U. But the rest of the letters, the rest of the bases are going to act exactly the same as they do in DNA replication. So we see a T here in the DNA, and we know that corresponds to an A. So we're going to add an A out of RNA next. And then a G is always going to pair with a C. So our next letter in our RNA transcript will be a C. And the enzyme that's doing adding on all of these bases, all of these RNA nucleotides, is RNA polymerase. So DNA polymerase was what we used when we were building a DNA copy. RNA polymerase is what we're using when we're building an RNA copy. So it's just going to be an enzyme that's moving along this template strand of DNA and adding the corresponding RNA bases as it goes along. So this transcript that we get, so here's our template DNA, here's our RNA polymerase. It's sort of moving along the template strand, adding RNA bases as it goes. And the one thing that I want to specify is that this particular RNA message that we're building is called messenger RNA. And that's because it's going to be sort of relaying the message from the DNA in the nucleus, and it's going to take that message out into the cytoplasm where the protein is actually going to be built. So it's a messenger, and it's sort of carrying the message from the DNA in the nucleus out into the cytoplasm. So the RNA copy that we're making is called messenger RNA. Okay, so again, here's this picture where you're taking the DNA original copy. It's going to stay safe in the nucleus, so we're just going to make a copy of it, a messenger RNA copy. We're going to allow RNA polymerase to go along one of the template strands and build a copy of it out of RNA, and then we're going to have our messenger RNA transcript that's free to go out and be made into a protein. So these last two slides are just emphasizing that there's one little step that has to happen after transcription. So we just talked about transcription going from DNA, making a copy of it into RNA. So that's in the nucleus because DNA is in the nucleus. But translation, which is the second sort of final process for protein synthesis, is out in the cytoplasm. It's happening at ribosomes, and that's going to be out in the cytoplasm of the cell. So RNA is the intermediary between transcription, because in transcription, we're going from DNA to RNA, and in translation, we're going to be going from RNA and making a protein. So RNA is sort of the end of transcription, but the beginning of translation. So it's going to have to go from the nucleus, where it's made in transcription, out into the cytoplasm, where translation occurs. So in between transcription and translation, our mRNA transcript has to move into the cytoplasm. So this next video will pick back up with this process at translation. Our RNA molecule will be in the cytoplasm, and we'll be ready to transform it or use it to build a protein. So I'll see you all then.